Hello everyone, it's my pleasure to introduce Accu16S/AccuITS today, accurate and broadly applicable amplicon sequencing for absolute microbiome quantification. Traditional 16S rRNA and ITS sequencing only provide relative abundance, which can easily lead to bias. Accu16S/AccuITS achieves absolute quantification using internal standard DNA. Relative and absolute abundance can be obtained with a single sequencing run, providing more ecologically meaningful analysis. The Accu16S/AccuITS workflow involves adding a synthetic spike-in sequence of known copy number after DNA extraction, followed by co-amplification with microbial DNA, and then using this to construct a standard curve for absolute quantification. A key innovation of Accu16S/AccuITS is the use of an ABI3730 gene analyzer to detect PCR amplification products via capillary electrophoresis, where peak area reflects molar quantity, allowing estimation of the spike-in ratio based on the peak area ratio of the target fragment to the indicator fragment. The main advantages of Accu16S/AccuITS include high accuracy, scalability, and broad applicability. This study summarizes 5 published examples demonstrating the broad applicability of Accu16S/AccuITS for absolute abundance quantification. Figure A and B applied Accu16S to investigate gut microbiota in glucocorticoid-induced osteonecrosis, showing results consistent with relative abundance analyses and confirming its reliability. Figure C showed that, in marine ecosystems, Accu16S-based absolute abundance closely matched transcriptomic data and revealed a decline in cyanobacterial abundance with increasing water depth. Figure D and E, F and G showed that, in fermented koji, Accu16S/AccuITS more accurately captured dynamic changes in bacterial and fungal communities, reducing compositional bias compared with relative abundance analysis. We have included 2 other case studies in the supplementary materials. In agricultural soils, Accu16S identified significant shifts in rhizosphere microbial communities under continuous potato cropping, as well as increased nitrogen-fixing and phosphate-solubilizing bacteria in 18-year decertified soils associated with Tamarix chinensis. Together, these examples highlight the robustness and wide applicability of Accu16S/AccuITS across diverse ecosystems. In summary, this study introduces Accu16S/AccuITS, an accurate and broadly applicable method for absolute quantification of microbiome amplicon data. By using ABI3730 capillary electrophoresis to estimate spike-in proportions from peak area ratios, it simultaneously provides relative abundance, total absolute abundance, and taxon-specific absolute abundance in a single run. The method is highly accurate, scalable, and widely applicable. Its analysis code is freely available on GitHub, along with an online platform for streamlined data management and absolute quantification analysis.